Before we get started, I have a quick favor. I've been self-funding the Finding Genius podcast for 5 years now. I've done over 3,000 episodes. And as you can see on YouTube, we're up over 1 million views on the channel, which is fantastic. The next thing I really want to push on is to get up to 10,000 subscribers. Because once we do, we'll be able to put a donate button and we'll be able to solicit donations to help keep the podcast running and to also get the Finding Genius Foundation moving along. We have a big project studying anxiety, depression, and PTSD and working on a product to help people overcome these problems because I've seen them explode recently after the last 2 years of the whole virus situation. So if you would, please subscribe to the podcast. That would help us tremendously. Give us a thumbs up and check in the description for Buy Me a Coffee. It's about $5. If you could buy me a coffee, I'd really appreciate it. It would help keep the channel going, and I love coffee. Thank you.
Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My guest today is Professor Sreetharan Kanthaswamy. I'm going to call him Sri for short. He is part of the School of Interdisciplinary Forensics. This is at Arizona State University, ASU. So I think it's going to be a great call. We're going to talk about the advances in forensics and the work that Sri does. So welcome, Sri. Thank you for coming.
Thank you for having me.
Yeah. Well, tell me a bit, how did you get interested in forensics? Did you watch like CSI years ago, or what inspired you?
Oh, this preceded a CSI show. I was interested in genetics, and I started working with various DNA analysis techniques. I was very interested in population genetics and conservation. And my first, foray into, this area was through wildlife forensics.
What's involved in wildlife forensics?
Conserving wild populations of endangered species. My area of interest at that time was orangutans in Borneo. So I was fortunate to get a small grant to help develop some genetic markers for understanding the social structure of orangutans in the wild, starting off with the paternity and or parentage, where we are also trying to figure out who the mothers are, and then using that information to understand their social structure based on their DNA. And then the markers that I developed became quite important to understand their population genetics, and also through that approach, we were trying to figure out if the populations were inbred, whether populations were being, animals from specific populations were being trafficked, and therefore you could use DNA to track the animals' origins and help repatriate them to those locations where they've been trafficked from.
Where were they being trafficked to?
They trafficked to other countries. There was a time when young baby orangutans were kept as pets. And
really?
Yeah, so that was one of the areas that
I know it's not a nice thing, but it'd be so amazing to have one as a pet, but probably not. They would probably
they're very cuddly, very cute animals. They're highly intelligent, and then once they turn about 6 years old, like about a human teenager's age, and I think your first instinct is to try to return them to their parent or to their natal population, and that's where the genetic markers can help repatriate these animals.
Did you get to spend time with them in the forest or in any situation? Have you hung out with orangutans?
Yeah, so part of my PhD work was collecting samples from these animals in the wild, and
that's so cool.
And also samples from from zoos, where they had these animals to help develop these markers, and then use these markers in populations from the wild. And
have you ever had any amazing experiences? Not like science based, but just human to orangutan experiences. Have you ever spent time with one and were just amazed by what you experienced?
They're highly, highly curious animals. They're very intelligent. They want to know what's up. So I remember once my family and we visited rehabilitation center, and this one female orangutan which was probably a subadult. She ran to us and she wanted to know what we were carrying in our plastic bags, and she inspected the bags like she worked for the TSA. So she was really curious about, what goodies she could find. And at the same time, we had another female orangutan who had a gibbon for a pet. So this interspecies interaction is pretty interesting.
Yeah, there was like Coco the gorilla that had a kitten for a pet
Right.
years ago.
Right.
That's really cool.
That's really cool. Yeah, they have a lot of interesting behavioral patterns that they share with us. Their genomes are highly similar to ours. One reason why we study them, because in many ways they mirror us in a more subtle and basic way that we can simplify our research experiments on them that we can't do on human.
You might be able to answer this. There's like, I don't know what you call it, an urban legend or something, but, that we share like 99% of our DNA with, gorillas or orangutan, whatever it is. Is that true?
Yeah, we do share a high percentage, probably close to 98 or 99% with chimpanzees, our closest relative.
But have you seen that by studying the genetics, actually? Has that been corroborated by your own experience?
Right. So we can actually see that high level of similarities. We've compared genomes. Other people have compared genomes in a wider scale and have seen very high levels of sequence similarities. But the question is, what genes are being activated in these various individuals as they are developing in utero or as they're developing after birth? Those genes, the timing of those genes being expressed is very important. Yes, we share 90%, 98% or 99% of our genomes. Sequence identity alone is not enough. It's what genes that are not shared, the small percentage of genes that are shared, that are not shared are the ones that potentially govern the differences that we are seeing in these animals and us. On average, humans share 99% of our genes, but yet you're a unique individual who's completely different than I am in many ways, in your appearance or your behavior, and so on and so forth.
Okay. So how did this come to the forensics part? Again, you were trying to determine if displaced orangutans were, like, where they came from, their provenance, how are you using forensics?
So you could use your DNA to figure out, just like Ancestry.com for humans, you can use DNA to figure out which population they were derived from, because they tend to have more relatives in that population that share a lot of genetic signal. It's just like parentage testing. You and your dad share 50% of your genes, and as the level of relatedness increases, as you start talking about your parents, your uncle, your first cousins, or your half-siblings, the level of shared genes start decreasing. But there are genetic signals that can help localize your place of origin, just like how we use Ancestry.com or 23andMe and try to figure out what our origins are in terms of geography. So the biogeographic signals exhibited by these genes are what we are using. I would not, maybe not the word genes, but genetic markers that we are using to figure out what our ancestry is.
Well, I would think the epigenetic profile would completely change, depending on local circumstance and hierarchy within the I don't know what they call them, groups of orangutans, what do they call them? A gaggle of geese, a pride of lions.
Yeah, so orangutans, I guess you probably call them a group of orangutans if you see bunches of them in one location. But orangutans are the only non-social primates aside, compared to humans and other.
Really?
The strongest social bond that you find is between the mother and the offspring until the offspring is about 6 or 7 years old when they are about to become, more autonomous. That's the strongest social bond. Otherwise, they are a solitary primate species, very different from a lot of other primates, including...
What does the male do with, where he mates with a female? Does he...
Right.
Do that and run away and the female takes care of him on her own, or what happens?
To a certain extent, the male is around to protect his access to the females. Females also guarding the territory in terms of the space and also access to food.
So are they like polygamous, where the male will have
the
females?
The males could have multiple females that they're breeding with, and females also can have multiple males that they can breed with discreetly. without the big male realizing that. So that can happen. And that's one reason why I use genetic markers to really figure out what is the degree of paternity among the males. Because there is that surreptitious mating that is going on. I mean, it's a way to shuffle genes and increase the gene pool by having extra pair mating, right?
Very interesting. But again, so I can see epigenetically they'll be very different now that they're in a new environment. But how do you figure out what's going on with the underlying genetic sequence?
And so another thing that may contribute to the differences, despite the shared similarity in terms of sequence identity, epigenetics also govern the regulation of some of those genes. But epigenetics, as the term denotes, it's changes in the expression of a particular region of the chromosome or the gene without an actual change to the sequence. So there's no sequence change. It's just a different pattern of expression based on chemical changes like methylation.
But that may give you a signal. Like if you were able to profile epigenetically a population of orangutans in Borneo, and then you looked at, one that's thousands of miles away, maybe epigenetically you could do a comparison, and that might tell you the provenance of the orangutan,
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
Right. Yeah, I mean, I would rely on the sequence identity more than the epigenetic changes, because epigenetic changes completely environmental or mostly environmental, and those changes are so spurious and it's hard to track. And this is not a controlled experiment, There might be. I mean, I've not tried this, and I folks, after listening to this podcast might be inspired to go down that path where they could use epigenetic changes to locate or to trace an animal's origin. But epigenetic changes take place generationally. They can take place when an individual moves from one environment to another environment. It's highly environmentally controlled.
Okay. So in terms of forensics, what else is all your forensic knowledge applied towards, again, figuring out, where displaced orangutans come from, or are there other projects you're working on?
Precisely. So I started off the forensic work when I dabbled with, I dabbled with forensic work as a PhD and a postdoctoral scholar. But I started working in a lab where they did non-human DNA testing for forensic purposes. And this is, when I say non-human, it was primarily animal DNA, where the topic of this podcast is supposed to be, try to get blood, saliva, or hair DNA. Animals have the same features. They have blood, hair, saliva, meat. So you could use animal evidence to help solve criminal and civil cases. So a lot of animal cases involve animals being suspects, where they may have bitten another animal or destroyed someone's property or attacked someone, whether they are pets or wild animals. Animals can be victims, they can be stolen, they can be harmed by people or attacked by other animals. And animals can also be passive witnesses, protagonists in a particular situation where animal hair can be used to connect a human suspect to another human victim, or human suspect to an animal victim, and vice versa. Find, secondary transfer evidence where the hair has been relocated from the crime scene to a different place, and trying to find links between these different victims or suspects or crime scenes. So animal evidence has been used in murder and other horrific cases that humans commit in others.
Like if they have a dog or if they have a pet or things like that.
So there's a very good chance if you went into a house with a dog or a cat, you're going to have hair evidence on your person. There's been studies that show that you're going to transport hair evidence. So, if someone can analyze that hair, they can actually put you in that particular location at some point. And it can be used to counter alibis, undermine alibis, or, things like that during an interview. Meat adulteration is another big concern. Animal trafficking, trafficking animals or parts of animals when they are illegal. They have databases for rhino horns and ivories and other animal parts. So they've used this to help combat wildlife trafficking as well. So from animal-oriented forensic work, I moved on to working with human forensic, and we contributed to building better DNA databases that represented the different ancestral or ethnic groups in the country or racial groups in the country. In the U.S., we also contributed to arguing that DNA databases, especially those involving humans, could be more race neutral rather than race specific. So we've tried, a lot of these types of work are done using research funds from federal agencies to help, shed some light on improving forensic approaches so that, forensic science actually contributes towards a fair and effective administration of justice.
What are some specific examples of various forensics techniques that have helped you solve, displaced orangutan or, some kind of crime scene? Let's, to our listeners, so it's not so nebulous. Like,
right.
What can we focus on?
So there have been cases where we found hair evidence from the victim's dog in the suspect's house, and comes one of those lines of evidence that we can place the suspect in the victim's house. We found contaminated food, with animal parts when it's supposed to be vegetarian. That could be criminal, but I think it wound up being a civil case with a hefty fine and, damages. These are a couple of examples. Animal cruelty happens a lot also. We have seen, victimized animals' blood on someone's clothing, and we can say, yeah, this guy or this person has that animal.
Literally blood on their hands, yeah.
Yeah, literally.
Okay. I know from shows like CSI and all that, people get a warped sense of what forensics really is about.
Mm-hmm.
What's, I don't know, like, are there new techniques that, they won't show in the shows, or, like, what is a typical forensic kit? What does it involve? What does it contain when you're looking at let's say a crime scene?
Right.
From your perspective, what are the main things that are looked at and usually?
Right. So the field of forensics is a very interdisciplinary field that tries to integrate different physical and natural science approaches. You have biology, you have anthropology, you have, physics, you have chemistry. So depending on what area an analyst or forensic scientist is an expert in or focuses on, it depends on, the tools that they use to help, solve or investigate and solve a particular case may vary. The whole idea is to use as much information as possible to help reconstruct the crime scene and, solve the crime. My expertise is in genomics and genetics. So I've used, I was trained in evolutionary and population genetics. So I use a lot of those techniques in trying to understand, how to solve a particular case, right? So when you're dealing with animals, for example, you have, unlike humans where you have one species and multiple different races, with animals you have many different breeds, right? They come from different environments. Their databases are not very, the genetic information on these animals where we compile and, construct databases is not very well established in most cases. So those are some of the challenges that we try to address. So we created one of the largest national databases for dogs because, every other household has one dog.
Amongst dog breeds, right? How different are they genetically?
Right. The dog breeds can be pretty extensive, but it's not just the breeds because previous databases focused on pure breed dogs because people use pure breed dogs for breeding and, all the other things that involve pure breeds. But one reason why pure breeds are very important is for research on them, scientific research on them, looking at traits or diseases and so on, behavior and so on and so forth. So they were a convenient set of samples for most people working at the university, and most animal forensic research is done at the universities. And they had, people working with animal forensics had access to these pure breed dog samples. But the catch is most of us don't have pure breed dogs. We have mixed breed dogs, right? Most of us don't pay so much money to buy an expensive breed as a puppy and raise it and be ultra, careful about raising it and so on and so forth. So most of us have mixed breed dogs. So one of the things that we did in our research was when we established this database, we made sure that there was a very good representation of mixed breed dogs in our database. And the database represented all of the U.S. and as many as the popular dog breeds at that time. So it was one of the first databases that were funded by a federal agency to help solve crimes.
What have you seen with the different dog breeds? Like how different or how similar are they genetically? How would you express that?
Right. So dog breeds probably developed in the last 200 or 300 years. So they don't have a long history, genetic history of being highly differentiated, but all of them have some level of relationship to their original ancestor, which is the wolf. And some breeds may not look like wolves, or may look like wolves, and they may not be as related to wolves as you probably assume. Like poodles, apparently, based on their mitochondrial DNA, are more closely related to wolves than German shepherds, which tend to look more like wolves, right, because of the features. So that was one of the things that we discovered, again, looking at a small section of the mitochondrial genome. People have come up with more interesting findings and better ideas of dog domestication and how they were derived from wolves in more recent papers.
So do things correlate phenotypically? So if I look at a Great Dane versus like a teacup Chihuahua, phenotypically or physically, they look radically different. But genetically, does that correlate? Has anyone looked at the variances amongst different dog breeds? And again, do they correlate in terms of difference? Like the more difference phenotypically the dog is, the more difference genetically the dog is, or is that not true?
Right. People have looked at relationships among the different dog breeds and come up with various types of conclusions. Dogs in general share very, very high levels of genetic identity, 99%. But it's those few genes, like I alluded to when we first started talking, that the few genes are the ones that control their shape and size and behavior. Right? If those genes are different between two breeds, then they are going to be different in terms of appearance or behavior and other aspects of their appearance or morphology or whatever it is.
Okay. So there's a few critical ones. And I guess, again, like a teacup Chihuahua versus a Great Dane, you'll see changes, but you'll see big differences in those.
When you look at those genes that control their shape and size, for example, you will see significant changes. But in other areas, they are not. And again, when we look at forensic markers, we are looking at neutral markers, typically markers that don't encode for any enzyme or behavior or trait, because we don't want any bias there. Same thing applies to humans as well. Humans, we use genetic markers that are considered selectively neutral. One, they're highly variable, very polymorphic because they're more neutral. And two, there's no, you are not, the risk of biasing the analyst is reduced when you use selectively neutral markers. If you're going after a suspect with blue eyes or dark hair or anything like that, there is a perception that there could be bias. But if you're using genetic markers that are neutral, then the chance, and you get so much information from those neutral markers, you don't have to worry about their appearance.
Like what kind of information do you get from these neutral markers?
You just like looking at the license plate of your car. It's just a series of numbers. Those numbers are unique. We are looking at over 20 different markers of a particular class of genetic markers. Actually, they're called microsatellites or short tandem repeats. These are highly variable regions within the genome, and they are inherited from your parents in a Mendelian fashion. So if there's a particular marker or allele your father has, there's a high chance that you would receive it from your father, and vice versa, an alternate form of that allele from your mother. And again, I'm not going to go into too much specifics because I probably need a whiteboard, but it's like, eye color, for example, right? If your parents are blue-eyed, the chances of you having blue-eyed, almost 100%. If one of them is brown-eyed and the other one is blue, depending on whether they're both those genes that control their brown eye color, brown eye, then the chances of you getting brown eyes, is very high. because the brown eye color is dominant. So we are looking at markers that are genetically inherited, that we can predict their inheritance. One reason why we stay away from epigenetics is, you really can't predict the level of inheritance without actually looking at the genes themselves and see whether they have a high chance of getting methylated, altered, chemically.
Well, now, has anyone ever looked into the epigenetic state of the parents before, at the time of conception?
I'm sure.
And then see how that correlates to the, I know that a lot of the marks are removed, but, there's prostosomes and things like that, extracellular vesicles that inform, the egg and the sperm and all that. So I just wonder how that would correlate. Has anyone ever looked into that?
I'm sure someone has looked into that because that's very interesting research. I have not, unfortunately.
Can't you look into everything? I'm just kidding.
Almost everything.
Okay.
But not epigenetics.
So do you feel like you're developing robust techniques?
Right.
How to, use forensics and genetics and meld them to figure out some of these things that you're figuring out?
Right. So we are using some of these things, and we are also teaching our students that in our labs and in our classroom, how do you learn from all these different techniques and find ways to combine these different techniques, so that you don't have like an investigative tunnel vision. Your mind is, you're utilizing all these different techniques. You can comprehend these different approaches that your colleagues in a crime lab is using, so that at the end of the day, you work as a team to try to solve a particular crime, right? So if a student comes to my lab, they learn how to use the most modern DNA techniques, and we try to prepare them for, jobs in the crime labs or any other science labs that require genetic testing or genetic analysis. So it's not just developing new techniques. Developing techniques, it's a hit and miss sometimes. Sometimes the progress is slow. Sometimes it's like, trips and spurs like. It depends on the funding situation. It depends on, luck to a large extent, luck. But we try to prepare ourselves, and the better prepared you are, the luckier you get sometimes, because, you tend to know all the different things that are out there that you can source from. And those are the types of thinking that we are trying to inculcate our students in,
Okay. So working in forensics, I mean, what is the romanticism part of it, and what is the reality of it? From working in it for so long, people that are interested in the career, let's say, anything you need to warn them of, anything you need to tell them that's unexpectedly good or bad, frustrating about it?
Right. I think we are all kind of like, as you pointed earlier, a lot of our students, a lot of us were inspired by shows like CSI. You're not going to be able to solve crimes in 45-minute segments. It's going to be painstaking, it's grueling, you have to be incredibly patient, you have to be highly collaborative, you have to be very, very competent, and be true to yourself and to your colleagues, because we make mistakes, and it's always better to own up to those mistakes rather than covering up. So those are the things that I think a lot of our students realize. You learn a lot of those things in your classroom, from grade school. But here you get to actually get some experiential training and things like that, and understand that a lot of these work is just hard work and dedication. We've learned from our professors, and they are learning from us. So this process of transferring knowledge and education will continue. So that's the best part about this. I really enjoy teaching. I enjoy the research. And I think my students see that passion in me. And I hope I can encourage them in the area of, becoming a teacher themselves, or becoming someone who works for law enforcement, particularly in a crime lab. So yeah, it's a job. And like any job, you want to do it as best as you can.
For a last point, I know we got to go in a minute, but what are some of the most common ways that forensic scientists make mistakes that can screw up a case?
Right. Not staying abreast with the latest technology, not being meticulous about their work, taking shortcuts, not re-reviewing their work, not having somebody else review their work, not working in, collaboratively with somebody else. Also, when they are rushed to solve something, that's when we make mistakes also. And also resource, right? Resource will help with the training, will help with the proficiencies, will help with the competencies, will help with the getting the best, most, advanced instruments, staying totally in line with the latest technology and understanding what that technology means, I think is places where I think We need a lot of improvement, but at the same time, being dedicated and being focused and always remaining competent by taking classes and staying abreast with the latest knowledge and technology is important. One other place that really hurts the crime lab is when they have backlog cases, especially from sexual assault cases. And there's this pressure on them to try to, go through these, sexual assault kits as quickly as possible with the least resources. That's probably one area where there needs to be a lot of improvement.
Okay. Well, very good. Sri, what's the best place for people to find out more about your work? And then for people that are interested in a career in forensics, where can they go?
Oh, yeah. They can Google Arizona State University Forensic Science, and that should take them to our school's website, and just a matter of, clicking a few buttons and figuring out what area, they might be interested in pursuing.
Okay. Well, very good. Well, Sri, thanks so much for coming on the podcast. It's been very interesting, and I appreciate the time.
Thank you so much, Richard.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed. Listening is your sole responsibility. Consult professionals when advice is needed.